Thanks, Sunny. Okay, so everyone should be able to see my screen now.
Yeah, you're looking great.
Get started. All right, thanks for having me today. I'm happy to be here. And like Sunny mentioned, I'm going to talk about fungicide resistance management, and we're going to do a bit of a deep dive into some fundamentals for the first little part of the presentation, and then a little bit more of a deep dive into what fungicide resistance really is. And this should build on if you were here earlier this morning and saw Dustin's presentation about anthracnose resistance, this should build on that. And then the last part is, how do we take that information, that guidance, and how do we put that into practice when building a program? And so we'll go through a short case study thinking about botrytis and anthracnose fruit rot. So if we go back to basics for when I taught plant pathology, we talked about the disease triangle all the time. And so you've got this titled Disease Management 101 because it really is a fundamental concept in plant pathology and managing diseases, where in order to have a disease, you've got to have three things. You have to have the right environmental conditions, a susceptible host, and a pathogenic organism. And so when we're thinking about disease management, whether it's fungicides or other tools, any method affects that disease triangle, okay, and how disease is produced. And I like to remind people that managing diseases is it's not just about fungicides, right? So in the presentation on anthracnose earlier today, you heard some really good examples of that. So there was some discussion about how different varieties of strawberries have different susceptibility to anthracnose. So that's one example, okay, variety selection. And obviously sometimes we're limited in what the options are because of the other agronomic considerations and market considerations. Okay, another example from this morning was there was a question from the audience about row covers and if that can spread anthracnose spores. So these are all other parts of managing diseases that are important to remember, okay, and can have an impact on disease risk. So getting into fungicides, and just some basics about fungicides that kind of have everyone here on the same page today or the same level. So fungicides are an essential tool to help you deliver high yields and high quality food in a sustainable way. Fungicides work best when applied before a disease attacks, okay, or very soon after a disease attacks. So I'm going to bring up today, I'm talking about preventative application, and this ties into that. So having the fungicide on before the disease attacks or the pathogen attacks. And then another important concept is that fungicides can be grouped into multi-site molecules that work on multiple pathways against a fungus to kill it, or they can be single site molecules that work at just one site. And that's a really important concept to understand when we're talking about resistance management. So what do fungicides do? So we will look at this kind of in four categories. So preventative fungicides, okay, when we use them preventatively, they work before the plant is infected. Okay. They are working before we see symptoms. These symptoms are the plant's reaction to being infected by a fungus. So we don't see symptoms until well after infection has occurred. Most fungicides work preventatively. And if fungicides work in multiple ways, then they work best when they're used preventatively. So having applications on, so there was a question earlier this morning about fungicide applied before rain, and that was good to hear in the sense that yes, you have the fungicide on before it rains, before that high risk period, before there's that high risk period of infection, right? So working preventatively. Some fungicides okay, work curatively. So this means that they stop the mycelial growth inside the plant. And this is happening still before we see any symptoms. And fungicides that have some curative activity are sometimes labeled as having kickback activity. So you might hear that term. Some fungicides are eradicant, but very, very few fungicides work this way. Okay? So an eradicant fungicide will stop the pathogen during lesion formation when we are seeing symptoms. We sometimes expect fungicides to work this way, but most don't work this way. Very few do. And then the last category are fungicides that are anti-sporulants. So this means they stop the pathogen from sporulating. So sporulating is what happens when that fungus is reproducing and producing spores. So essentially producing like babies. Okay, then the fungicide is stopping that process. Okay, so we still see symptoms at this stage. Lots of fungicides work this way, but it's important to remember that your crop is already infected, okay, when that anti-sporulant activity is happening. So throw up this visual where we have this preventative, curative, eradicant, anti-sporulant across the top. And then down here, we have the processes of infection, okay, and within the plant to symptom development. So you have these green parts are a healthy plant cell, okay, and the orange is the pathogen. So initially it's on the leaf surface, okay, then it's infecting, getting inside the plant and growing inside the plant. Then the lesion is forming and then it's producing spores. Okay, so you can see here preventative fungicide is working at this stage, okay, before infection is occurring. Curative, we don't see symptoms yet. Okay, eradicant, it's stopping this mycelial growth within the plant. Okay, and then anti-sporulant, it's affecting this stage where new spores are being produced to spread disease. If we shift the focus a little bit to important characteristics of fungicides, we can break it down into 5 key categories. So the mode of action, so that's how the fungicide affects the pathogen. Okay, spectrum is what it works against. Okay, which fungal species is it effective against? The physical and chemical properties, so how does it move in the plant? Okay. The length of control, okay, and then the potency, how much is required. We're not going to talk about all of these today, mostly about mode of action, okay, and a little bit about spectrum. So mode of action, okay, what does that mean? So here's an example from the Moravis Prime label. It has 2 active ingredients in it, okay, pidiflumethafen, okay, also known as adepidin, and fludeoxonil. So I've broken down here into these rows. We have something called mode of action, site of action, a FRAC code, and the group name. So the mode of action, okay, that's the process that's impacted in that fungus when that chemical is applied. So for pidiflumethafen, it's respiration, fungal respiration, and in fludeoxonil, it's signal transduction. Okay, and then the site of action is like where that's happening. Okay, so these 2 things, as if you're an agronomist or if you were a grower, these things might not be of primary importance for you to know or remember, but it's important to just think about that, the concept of that mode of action, because you'll see that they're different, okay, between different active ingredients. And it's this mode of action, okay, that helps determine the FRAC code. So the FRAC code, we'll talk about FRAC in a few minutes, but when we talk about alternating or rotating modes of action, we're talking about rotating between different numbers, and sometimes the FRAC code is a letter. Okay, so for multi-site fungicides, like if you see an M, okay, that's a multi-site, but that's a FRAC code. So knowing those FRAC codes is really important. And then each FRAC code is associated with a mode of action group, and there'll be different group names. And the group name can be based on a number of different things. So sometimes it's the chemical structure, sometimes it's the site of action. So with group 7s, we often refer to those as the SDHIs, okay? With group 12, PP or phenylpyrroles, for example. So key things to remember from this slide, FRAC code is important to know, and then getting used to these group names. So mode of action of some botryticides. So this is just an example of some different botryticides. They're not necessarily all labeled on strawberries, and that's not all inclusive of everything that's labeled on strawberries, but to give you an example of what that mode of action, okay, means. So we've zoomed in here on this animation into a fungal cell. Okay, so pretend this is under a microscope. And then you've got these different organelles in the plant cell doing different functions for that fungus. And then we've got these different modes of action. So we have SDHIs, so they're affecting respiration. So you've got that little arrow pointing to where respiration happens in the cell. Okay, you have, and so that's something like Circatus fontellus, for example. Timurex gold is affecting lipid synthesis. Okay, AP fungicides, they're affecting another component. Okay, PP's another component. Okay, Diplomat affecting another component. Okay, and so on. And then you have your multi-sites, and those are affecting multiple functions within that cell. So single sites versus multi-sites, right? And so based on that mode of action, you've got different group names assigned here. So group 7s, for example, and just to point out, some products will have more than one active ingredient. So you've got Moravis pine here as a group 7, okay, and as also contains a group 12. The coloring on here helps to denote resistance risk. I won't dive, I'm going to dive into that in the next section. So fungicide resistance. What is fungicide resistance really? Okay, so we can pull up the definition here. Fungicide resistance, it's the acquired heritable reduction in sensitivity to a specific fungicide. So it's something that is acquired, okay, and it will pass from generation to generation. So it's inherited. There's something called lab resistance. Okay, and this is where populations are in a lab and they're artificially mutated to lose sensitivity to a chemical. That's really of academic importance. What is of importance, okay, in agriculture is field resistance. And this is where we have changes that occur within a specific fungal species under field conditions. So out there in commercial fields, okay, the first indication that this might be happening is through routine monitoring. Okay, or okay, if it's later on in the development, if we start to see poor performance of a specific mode of action group. Okay. One thing to keep in mind is that a performance failure doesn't always mean that there's fungicide resistance. So there's other reasons why things can go wrong in terms of why something didn't work. So it's important to consider as a possibility, but it's not the only possibility. And resistance is a concern in Nova Scotia. So we saw a presentation earlier today about that. Syngenta is also doing work in Nova Scotia to better understand botrytis and anthracnose resistance. So we worked with Sunny last year to collect some samples for testing as part of a national project, and hoping to do that again this year. So we don't have results to share on that today. Once we get 2023 samples in, everything will be tested together, and we look forward to sharing that back in the future. So we talked about what is resistance. Okay, we know it's a concern, but how does it really develop? Okay, so we'll go through 3 scenarios here. We've got scenario A, where we spray with a single mode of action. Scenario B, where we are alternating between modes of action. And then in scenario C, we're applying mixtures of modes of action. So we'll go through each of these things and some animation. And when I'm going through these, resistance happens as a result of mutation, okay, recombination and migration of the pathogen, and then selection pressure on that resistant population. So we look at this, we've got this, hypothetical population on here. So we have all these little triangles, okay? We have a population of 16, so n equals 16. We have mostly green triangles, and then we've got this one golden one, okay? And that's representing our naturally resistant population, so 1 out of 16. So that gives us a resistance population of 6%. So we go along and spray a fungicide, okay, with mode of action X. And we're selecting, okay, for a resistant population. So fungicide killed most of the green ones. Okay, it didn't kill that yellow one. And so now instead of a 6% resistant, okay, we have 1 out of 4. So we have a 25%, okay, level of resistance. Now we have our multiplication stage. Okay, so we've multiplied now our population is at 8. Okay, 2 out of 8 are that golden color. So now we still have 25% resistance. We spray again with the same mode of action, and we select again for those yellow triangles. So now we have 100% resistance. And then that population multiplies, and they're all yellow. So this is a simple example to demonstrate repeated use of the same mode of action accelerates that resistance development. What if we alternate between different modes of action? So we're loading up the same scenario at the beginning here, where we've got our circle with all these triangles. We have 16, there's 15 green ones that are susceptible to the fungicide, and then that one golden one. So we're at 6% resistance. We apply mode of action X. So we're selecting, okay, for the golden triangle. So now we have 25% resistance. That multiplication step happens, reproducing. We're still at 25% resistance. So we spray again, but this time with mode of action Y, okay? And the golden triangles are susceptible to mode of action Y. So alternating modes of action reduces that risk of resistance development. The last scenario is if we apply a mixture of modes of action. So we're back to our same starting population. We've got 16 triangles, 15 sensitive green ones, and 1 resistant gold one. We spray X and Y. We've got a couple individuals left. Okay, we're at 50% resistance here. Okay, 50% gold triangles. We multiply. Okay, so we're still at 50%. But then we apply, our mixture again. We respray, okay, and this time those individuals have been killed. So when we combine active ingredients with different modes of action, we reduce the risk of resistance development. There's also different types of fungicide resistance, okay? And we can categorize them as destructive or continuous. So with destructive resistance, I'll try and explain to you this graph. We have, the sensitivity of the fungus down here, and then on the vertical axis is the frequency, okay, of individuals. Okay, so the higher, okay, the more, this means, most of the population here, and this red line is in this green zone, so in the sensitive zone, okay, and this is in year 1. And they're looking at this over time, okay? So you'll see that in year 1 and year 4, the yellow, the red and the orange most of the population is sensitive. It's in this green zone. Okay, there's a few individuals that are in this kind of red zone, this resistant zone. But then we see this big shift year 5 and 6, where there's fewer individuals in that greenish zone, and then a lot of individuals, okay, especially in year 6, in that sensitive zone. And there's a big gap here, so there's been a big jump. Okay, and so this resistance, okay, it's called disruptive or strong resistance, sometimes qualitative resistance, where all of a sudden, okay, the population is insensitive. And these are some examples of products where we see disruptive resistance. So Quadris would be one example to kind of link to the presentation earlier today. Is also a group 11 fungicide, and typically what we see with group 11s is this disruptive resistance where it just no longer works anymore. The other type of resistance is continuous. So sometimes this is called polygenic, okay, or there's a shift. And in this case, we see a gradual shift in sensitivity over time. So again, down here, you have your sensitivity. So this is how much chemical is required to reduce growth. Okay? And so you have this kind of green zone moving into a red zone. Initially, that population in the red line is pretty sensitive. Most individuals are pretty sensitive, but then over time, you require more and more chemical, okay, to reduce growth. And so group 3 fungicides would be an example of a group where we see this continuous resistance. And then Moravis, so this is an example of a group 7 fungicide. It's a little bit in the middle because there are a few cases where we see disruptive resistance, but it's mostly continuous. All right, so I mentioned FRAC earlier and said I'd tell you what it was. So here we are. So FRAC is the Fungicide Resistance Action Committee. And FRAC is a specialist technical group of CropLife International. And what they do is provide guidance on fungicide resistance management to try and prolong the effectiveness of at-risk fungicides and also limit the crop losses should resistance occur. They work in a number of ways, and one of them is they have several working groups where they are typically linked by fungicide mode of action group, and they'll talk about what the guidelines should be, so for individual groups or modes of action. One of the things that is considered when determining fungicide risk, so there's actually 3 things. So it's the fungicide risk, the pathogen risk, and also the agronomic risk. So fungicide risk is the inherent risk of the fungicide, okay, to develop resistance. So you'll see that FRAC has classified different modes of action groups into different categories. So you'll see the group 11s up here and high risk. The SDHIs, or the group 7s, are considered medium high. And then you have others grouped in medium. So multi-sites will be low risk. For the pathogen, they are also classified from low, medium to high risk. You'll see botrytis here considered high risk and anthracnose medium risk. And then the agronomic risks. So these are the other production factors. So, you know, one example, we talked about those examples, like other ways that you manage diseases at the beginning of the presentation. So, you know, variety selection, row cover use, you know, if you're using overhead irrigation or not. I use the crop rotation as an example in a lot of annual crops, but it doesn't apply as well to strawberries. So there's increasing risk, okay, when there's higher pathogen risk, fungicide risk, and agronomic risk. When we have increasing risk, FRAC tends to tighten up the recommendations. So I have 3 examples here with group general recommendations for group 3, 7, and 11 fungicides. And you'll see we're going from medium to medium high to high risk. And when you look at the recommendations for if these should be applied solo, if they should be alternated, if they should be applied in mix, and how many applications can be made, they get more restrictive as you move into the higher risk. So an example here with group 3s, solo applications fine, whereas with group 11, rarely would solo applications be recommended. You also see a limit on the total number of applications, the number of applications of a group 7 or group 11 containing fungicide as a percentage of the total number of applications in that program. So why is mode of action important? I think I've emphasized this already, but for resistance management, we need to know the mode of action group. There's different rules for each FRAC group. In general, when targeting the same disease, we want to mix or alternate with a different group. If you do not know where to find the FRAC code, the easiest place to find it is on the product label. On the first page in the upper right-hand corner on Canadian labels, it will always show you the FRAC codes. So this is an example from Ravis Duo where you have a 7 and a 3. We often get questions about, is it better to mix or is it better to alternate? Both mixing and alternation help dilute the selection pressures. If you think back to those animations I showed you, those 3 scenarios, we're reducing that selection pressure on resistant populations using either way. When we are mixing, okay, if it's a premix, it ensures that the resistance management strategy is implemented. But with alternation in multi-spray crops, where we're applying multiple applications in a year, it can start to get a little bit confusing when everything's premixed, because you have different modes of action and you're trying to alternate and do all the right things. So sometimes with alternating, it's easier to keep track of the mode of action versus a premix. In general, mixing is the best practice when there's fewer applications, but alternation is considered to be okay when there's many applications made in a season. So a lot of horticultural crops. A watch out here with fungicide premixes is that they can serve 2 purposes. So they can be built-in resistance management, but sometimes there are premixes that only serve to increase spectrum. So they're built-in resistance management when both of the active ingredients in the mix are effective against the target pathogen. If the active ingredients in the mix are targeting different pathogens, then they're not a resistance management tool in that case. So not all mixtures are good resistance management tools. We've covered some plant pathology and disease management basics, and we've covered some fungicide resistance basics. So how do we take that information and put that guidance into practice? One way to think of this is if you go back, if you're aware of the 3 little pigs story, where you have pig 1 builds a straw house, pig 2 builds a house made of twigs, and pig 3 builds a house made of brick. The big bad wolf can blow down the straw and the twigs, but not the brick house. So we're thinking, how do we plan and how do we build a brick house for resistance management purposes? So some fundamental things to keep in mind. Remember the beginning that managing resistance is not just about fungicides. So those other things that you do for integrated disease management also affect resistance management. General approaches: apply fungicides preventatively, follow the label application rates, and ensure you've got your sprayer set up to get proper coverage. And that could vary with growth stage and time of year, canopy coverage, etc. So thinking about carrier volume, thinking about nozzle selection, and those kinds of things. Now, specific to the program, try to follow the guidelines from FRAC working groups. So this can mean limiting the number of applications in a mode of action group per season, or how many times something is applied consecutively. And this can be pathogen or crop driven. And we see that a lot with botrytis, for example, where there's botrytis specific recommendations, or in crops that are multi-sprayed like strawberries. Alternate or mix FRAC mode of action groups that are active on that same disease. Okay. And incorporate low-risk fungicides, like fungicides that are group M and BM. So BM is biological multisites. I'm going to go through a simplified case study to try and illustrate this. Okay, how do you start planning and how do you work out a plan for this season? And I'm going to present it in this way, thinking about what you need to know when you're planning. So know your diseases, know your tools, know the rules, and know your local risk. And you use this information to plan and build your program and build that brick house. So first, know your diseases. So we're talking today about botrytis and some basics, okay, on biology for botrytis. You know, it's ubiquitous in the environment, overwinters on residue, and it has a wide host range. So that's why I'm saying it's ubiquitous in the environment. We know blossom buds and flowers are particularly susceptible to infection, and that it's favored by moderate temperature and high humidity and moisture. So I've drawn out this kind of relative risk curve here over the growth stages, and this example is in June-bearing berries, where we start to see disease risk kind of increase, okay, into that bloom period. And then anthracnose. So focus for anthracnose for me today is on the fruit rot stage. So that's most commonly associated with Calototrichum acutatum, the species complex there. So it can overwinter on residue. And as was mentioned earlier today, we saw the other presentation that on anthracnose, favored by warm to hot and very humid conditions, and that's splashing, especially during bloom and harvest. So that's what we're dealing with on the disease side. Know your tools. So I'll pop this up. Got all these boxes, and I listed modes of action for products labeled for botrytis or anthracnose control or suppression in June-bearing strawberries. So I pulled this up from some online resources, and I've just organized this by group, mode of action group. And it's kind of a big mess. It's hard to understand. Got all these modes of action. What do you do? And so what I suggest is then you take that information and you start to organize it into what's labeled for botrytis, what's labeled for anthracnose, and what's labeled for both diseases. So you can take those groups and organize them that way, and starts to give you some idea of what you have to work with, focusing on botrytis or anthracnose or both. And a reminder here to look at specific product labels for details on target pests, because some of the products that have the same mode of action group might only have one or the other on the label, and it will vary if it has control or suppression on the label. Know your rules. This means reading and adhering to label directions, and remembering that not all directions are related to resistance management, but they're still required, and they can impact product fit. So some examples: you're limited to 3 applications of an M5, so chlorothalonil in strawberries. You need to consider the restricted entry interval. Is it 1 day? Is it 6 days? Is it 30 days? And the same with PHI, right? Because that will impact where things are placed in the program. And there could be other secondary standards that you have to deal with as well. And then know your rules. So I showed a slide like this earlier, like as a general example. This one is more specific to botrytis and anthracnose in strawberries. And I've highlighted group 7, 11, 9, and 12 here because these are kind of the primary single site mode of action groups with medium to high resistance risk. So I've listed here, yeah, can you apply solo? Do you have to alternate? Can you apply mixed? How many applications can you make? And in an 8-spray program, because that's what I'm using for our case study example, how many applications does that mean? So I'll focus down here on number of applications. You can see it varies, but it's never more than half. Okay? And this variation, it depends on if it's applied solo, okay, or in a mix. And what this means in an 8-spray program is that for 7s, 11s, okay, 9s, and 12s, we're basically limited to 3 applications or less. 4 for group 11 if applied in a mixture. The last bit of information to help you plan and build your program is to know your region and support local monitoring programs to inform that decision making. So example would be the project that was presented earlier on anthracnose resistance. And then we're also trying to build that knowledge with our botrytis and anthracnose monitoring program as well. So let's look at this example. I'm going to build, I've gone back to the strawberry growth stages over time here. And then we have that relative risk of botrytis and anthracnose, which is hard to draw, right? Because we've talked already today about how the weather has such an impact on disease risk. And it's back to that disease triangle, right? But for purposes today, we'll take this as the relative risk. And so in general, first step, we this preventative pre-bloom sprays, okay, focusing primarily on botrytis, and you could put in your multi-sites there. Then as risk starts to increase, getting into that bloom period, we put in a single site with 1 or 2 modes of action and a multi-site. This may be the last opportunity to use a multi-site or get into harvest, and those PHIs and REIs become restrictive. And then implementing a preventative fruit rot program during harvest and fruit development. So what does this, what this could take it to the next step? This could be like an M5, M5, M4 early on. So this is chlorothalonil and Captan, for example. Okay, and then we've got those single site mode of action integrated into the program as risk starts to climb. And then in that fruit development and harvest period, we're looking at single sites. But what we're doing here is trying to integrate group M's, okay, and BMs, you see here, and M's here. We're looking at group 7s, 9s, 11s, and 12s, okay, and trying to limit those to 3 or fewer sprays. So I can show you some counts here. We've got a 9, 9, okay, we have a 7, 7, 7, so that's 3, 12, 12, 12, so that's 3, okay, and in this example, there's just 1, 11. And I should say this is not a prescription, okay, but it's an example of how to plan and how to work in. Okay. Another check, are we alternating and mixing single site modes of action? So we're definitely mixing our single sites modes of action here, and then looking at alternating, trying to separate the group 7s, okay, so they're not back to back, trying to separate the group 12s, so they're not back to back, separating the group 9s, okay, so everything here is separated and not back to back. And then also considering spectrum. So in this example, we're assuming that botrytis and anthracnose are both a concern, especially later in the growing period. Okay, we're thinking about getting into warmer conditions, and then that anthracnose risk starts to increase along with the botrytis. So the boxes with green and yellow mean that we're covering the spectrum for both, okay? So how do you build a brick house to delay resistance? Review here. So think about the fundamentals, back to that disease triangle, okay, host, pathogen, environment, and integrating management techniques and not just relying on fungicides. Those fungicide fundamentals, mode of action, in this case, we really talked about resistance fundamentals and what's affecting risk and what is the risk for your particular diseases. It's fairly high in for botrytis, right? And anthracnose is not far behind. Implementing the resistance management guidelines and understanding that those will change depending on crop and pathogens sometimes. And knowing your diseases, the tools, the rules, and the region, and it comes down a lot to planning, okay, and working through some examples. And that's what I have today, and I'm happy to hear what questions you have.